Okay, class, where we are now, we want to talk on, first of all, there's several subjects in this video, but one is the blood-brain barrier. Now, let me kind of explain. Neurons are very sensitive. As you know, they don't do mitosis after a certain age, so you cannot afford to lose them. In the blood are so many noxious substances, waste, and everything that's being transported to be eliminated by the kidneys or other things, that you cannot afford the delicate neurons to be subjected to that. So therefore, the blood vessels in several areas of the brain, not the whole brain, are protected by something called a blood-brain barrier. The way the blood-brain barrier is, here is the blood vessel epithelial cell, which you'll learn in ENP2 is called an endothelial cell. Well, what happens is that here would be the lumen where the blood travels. So this is the epithelial cell. So surrounding this are one of the neuroglial cells. Remember, microglia, astrocytes are astrocytes right here. And what you have here to separate, so this picture here shows the endothelial cell. Here would be the blood, separated by using these astrocytes, and the other thing are tight junctions. The endothelial cells are locked by tight junctions, continuous capillary walls, in order to kind of prevent that. So astrocytes and tight junctions. So protective mechanisms help maintain a stable environment. Blood bonds are separated from neurons by continuous endothelium, tight junctions, relative thick basal lamina, which we'll call basal lamina, and also bulbous feet of the astrocytes we were talking about, right? So that's the blood-brain barrier. Okay. Selective barrier that allows nutrients to pass freely. Okay, it's ineffective against lipid-soluble substances. Let me say that again, because lipid-soluble substances can pass through cell membranes. That's why that poisons, when I say poisons, I mean things like insecticides, should be water-soluble, because you have even the skin doesn't. Absence in some areas. Okay, some areas, the neurons need to experience what's going on in the body. For example, in the vomit center, in the hypothalamus. These areas need to be able to experience so they know what to do. Stress, because of the mobilization of some white blood cells and some other things, can cause damage to the blood-brain barrier, continuous stress. All right. Now, we go further then. So now we're to something else. We're talking more like now about brain anatomy, okay? The cerebrum, which is the upper part of the brain, this is it here, the cerebrum. See, the brain kind of caps over everything. So the cerebrum grows and sort of caps over everything, okay? And what I mean is, if I can find it, see, right up here is going to be, in this prosencephalon, is where you're going to get that cerebrum developed, which would be this. See, those hemispheres trying to come out on you, that telencephalon. And this kind of forms a mushroom kind of thing over the rest of the brain, kind of a mushroom thing over the rest of the brain. So assume this right here is kind of like a mushroom, I'm going to call it that, over the rest of the brain, which would be underneath. That's the cerebrum. That's the biggest part of your brain. Forms the superior part of the brain and makes up 83% of the mass. Contains ridges and grooves. Okay, so let's look at this. The ridge like that is called a gyri. Okay? Gyri. Singular would be gyrus. So all of them are gyri. These little shallow deals are called sulci, or one is a sulcus. Anything a real deep cut like this, deep groove, is called a fissure. okay, are called a fissure. Now this right here is your longitudinal fissure, which is separating the right side of the brain from left side. Okay, now the reason for these gyri and sulci, you may recall, is that the brain needed a lot of surface area and you didn't want to make the head any bigger. So therefore, the skull didn't come out, so it forced these foldings of the brain to keep surface area without getting a bigger skull. Okay? So this is what you're looking at. All right? So this is an anterior view, posterior view, lateral, and so forth. Okay? And superior. All right, so we go further. The deep sulci divide hemisphere into 5 lobes. So the cerebrum has 5 lobes. See if I can, if we go here. It has a frontal lobe, which would be under the frontal bone. Okay. It has a parietal lobe. It has an occipital lobe. Okay. Then it has a temporal lobe. Then one lobe is buried within the confines of the brain, and that's called the insula. The insula. This right here. So frontal, parietal, temporal at the back. Occipital, I'm sorry, temporal. Occipital at the back, temporal on the side, your temples, and then this insula down here. Okay. This right here is called the central sulcus. And it goes straight across the brain. Straight across the brain. And it separates the frontal lobe from the parietal. The frontal lobe from the parietal. Okay, so we come here. That right there. Then see right here was that that primary motor cortex we talked about in that last one. And that primary somatosensory cortex. Okay. So the precentral gyrus, the ridge in front of the central sulcus, is under a functional thing, the primary motor cortex, and this is the primary somatosensory cortex. If we look at it on a functional, we look at it on anatomy, it's called precentral gyrus, postcentral gyrus. We look on a function, it's called primary motor cortex and primary somatosensory cortex. Okay, so here's that picture again. Okay, let's see what we got. So we have a parieto-occipital sulcus. Parieto-occipital sulcus. That's back here. Parietal, which is separating the parietal from the occipital. A lateral sulcus, which would be up in here, that separates the parietal from the temporal. And the precentral and postcentral, as we alluded to, surround this central sulcus. So we had the central sulcus, the parieto-occipital sulcus. So the central sulcus separated what? The frontal lobe from the parietal. Separating the from the parietal, separating that from the occipital, is the parieto-occipital sulcus. And separating the parietal and really the frontal from the temporal would be this lateral sulcus. Here again, the humps of gyri, the deep things, the little light, shallow areas of sulci. Okay. So the cortex, we said, which was this part here. Let's see if I think I had another one, didn't I? The cortex, which is that part there. See if I can find another good picture. Which is the bark of the whole thing, that cortex. Let's see where we got something. That cortex right there. Ah, here's my picture. This cortex right here is gray matter. This is where you have cell bodies. Cell bodies. Counts for 40% of the mass of the brain. That's that cortex. That's where you have the cell bodies you think from. It enables sensation, communication, memory, higher level stuff. Each hemisphere acts contralaterally. We already talked about that because we did the spinal cord. Controls the opposite side of the body. Hemispheres are not equal in function. Right more artistic, you're more right side dominant, left more technical, mathematics, science. No functional area acts alone. We have found that if you think about something or do a math problem, there are many areas that light up. There's no one area. We found this later. We'll talk about that a little more. When I say light up, if we do a certain test, certain scans of the brain, then we can see that to happen. Okay? All right. So we go here then. What we have is three types of functional areas, generic functional areas. Motor areas, which control voluntary movements, sensory areas where you evaluate things, and association areas. Now, we talked about motor areas. Well, we talked about at least part of the motor areas, not all of them. We talked about the primary motor cortex. Remember the primary motor cortex. Let's go back, which was up here, the primary motor area. And then we talked about the primary somatosensory. But there are more areas than just that. That's the primary one. Then we have these association areas we'll talk about. Okay. Several anatomists attempt to find physiology of this. K. Brodmann in 1906, wow, he did something. He found 52, see these numbers, functional areas, thus numbered. Now, later we found out by doing certain scans, that several areas light up, not just Brodmann's area. But one thing is amazing. Without all of the modern technology, he was able to determine what lights up the most. So no one area does it all, but there is an area that's more involved in it. And that's what he really found with this particular aspect here. Okay. So we go further, and that would be what he was showing there. Here again are some other functional areas splitting the brain in two, some of his 52 areas. There. And so here are some areas that he found. The primary motor cortex, which was right here, that precentral gyrus, that's 4, the primary somatic cortex. The premotor cortex, which is in front of that, we'll talk about. Now, the primary motor cortex, which you saw before, that's where they had that little man, the homunculus. That primary motor cortex controls your skeletal muscle. But the premotor cortex tells the skeletal muscles what to do. So let's say you're learning to play the piano. Then the premotor cortex neurons there would tell the primary motor cortex which fingers to move. The primary motor cortex would move those fingers. Then we have Broca's area, Broca's speech area, which is a Brodmann area, 45 and 40. Brodmann also did some work, in case you're trying to figure out what. Brodmann did the areas, the 52, and then Broca came in and did some other work. So in other words, when we spoke of this, let me go back, we said that those functional areas were determined, Brodmann, 1906. Okay, then another anatomist, Broca, came in and did some work too. So we're going to start right here at those functional areas again. We'll close this video